Good morning, everyone, and welcome to this fantastic venue. Montreal is such a beautiful city. I used to live in Ottawa. I was there for a couple of years, so it almost feels like coming home. So firstly, I want to thank the organizers for inviting me to speak here today. It's indeed an honor and a privilege. And I want to thank all of you for being present and listening. So my name is Dave Butler. I'm the CTO at Hongene Biotech Corporation. And today I'll be talking about the evolving landscape of oligonucleotide manufacturing and meeting future demand with ligation technology. So again, a bit of a change of gear. Disclosures: I'm a full-time employee at Hongene Biotech Corporation and also serve on the board of Akte Therapeutics. So for an overview, I'll talk about the evolving market for single-guide RNA and siRNA. And these modalities really represent the lowest hanging fruit for ligation and also the biggest need. 3 generations of oligonucleotide synthesis technology, mechanisms, properties, and immediate applications of ligation. Examples of products that we've synthesized by ligation at Hongene. CMC features of ligation and current and future oligonucleotide fragment manufacturing processes. And then I'll finish with future directions, conclusions, and acknowledgements. So it's good to start by taking the pulse of the market. And this data was collected from the Beacon database with the kind help of the folks at Hanson Wade. But firstly, gene editing drugs. And since their introduction to the clinic in 2017, there's been really robust growth in this modality. There's been 17 drugs that have entered the clinic so far in 2024. The first approval, of course, last year, which was Casgevy. And over this period of time, we've seen the introduction of 3 unique gene editing modalities. And that's those 3 on the bottom. And this rapid evolution of gene editing modalities creates formidable challenges for oligonucleotide manufacturers who are asked to manufacture ever longer and chemically more complicated single-guide RNA molecules. And these are expectations that currently stretch our capabilities to the limit, as we'll see. Switching attention to the siRNA field, and 82% of clinical siRNA assets target at least one common disease indication, and that's defined as affecting more than 1 in 2,000 patients in the US. Of the 307 indications in the database for siRNA, more than a quarter of them are connected in some way with cardiovascular disease. And this focus on cardiovascular disease creates a huge market potential for siRNA drugs. These 6 disease areas in the table, each of those has the potential to reach about 10 million or more patients just in the US. Inclisiran has already been approved, and there's the potential for 3 more approvals in these disease areas by 2030. And if we do a back of the envelope calculation and say that 1 gram of each of these siRNA GalNAc conjugate siRNAs would be required to treat 1 patient per year, then 10 million patients needing any one or a combination of these drugs would create a market demand for 10 tons of API. And by our reckoning, that would stretch current global capacity to its limit. And if we look at this graph on the right-hand side, this is total FDA approvals for monoclonal antibodies and siRNA therapeutics. And monoclonals have about 25 years head start on SI, and it could be argued that they're going through their precocious teenage growth years in terms of their life trajectory. Whereas siRNA therapeutics are really still in their infancy with 6 approvals so far. And really all of this potential has yet to come. but all of the indications are that it is coming. And as a field, we need to be ready for it, and the manufacturers in particular, we have to make sure that we can get these drugs to the patients who need them. And so we find it useful to think about oligosynthesis technology in terms of 3 generations. Firstly is the very well-known chemical synthesis cycle, also known as the phosphoramidite method, that has been the mainstay of oligonucleotide development and manufacturing for the past 40 years. Next is generation 2, which is the ligation technologies, and we'll go into much more detail about that on the subsequent slides. Then finally, at the bleeding edge of oligosynthesis technologies is fully enzymatic synthesis. And in this approach, enzymes like polymerases and TDT are used to assemble oligonucleotides in a way that mirrors the way that biological processes do it. The monomers in these cases are NTPs generally, or 3' protected NTPs. So looking at these different generations together, the first thing to note is that chemoenzymatic ligation processes currently rely entirely on generation 1 technology. So phosphoramidites are required to make the oligo fragments that are needed for the ligation. But this is good because it means that ligation technology is essentially ready for manufacturing today. The product purity and yield are higher for ligation chemistry, and we'll go into the reasons why that is. Process mass intensity, that's a measure of environmental sustainability. That's 3,000 kg of raw materials are required to make each kg of API for generation 1 processes. Actually, in excess of 3,000, that's the lower end. Whereas for generation 2, by virtue of the fact that the yields are higher, and we're also partly in an aqueous system, the impact on the environment should be lower. Generation 3, meanwhile, the development is still very early. You know, the supply chain for NTPs, the raw materials, that all needs to be figured out. But there's no barrier in physics or chemistry that prevents the engineering of enzymes and chemistry and processes to one day provide material through this method that with unmatched purity and yield and PMI. But I've left these boxes as orange for now, simply because it's, you know, there's still some way to go to actually prove that. There's been a slew of publications in this area over the past year or so that really highlight the promise and potential of this technology. So taking a bit of a dive into the mechanism, first of all, the ligase, a lysine on the ligase reacts with ATP to generate this adenylated species that then undergoes adenylyl transfer to the 5' monophosphate. This bridge to diphosphate intermediate that results can then undergo reaction with a 3' hydroxyl on the adjacent oligo fragment strand, with loss of AMP and sealing of the nick to generate the ligated oligo product. And through this reaction, there's this reaction mechanism, there's some important properties that result. Firstly, some of the shorteners are rejected during the process simply because they're not substrates for the ligation reaction. They're too far away from the nick site. Then specificity, yield, rate, these can all be optimized through changing the process conditions. And specificity is generally very high for these enzymatic reactions anyway, so we don't see impurities that are different from the ones that you would see in regular oligonucleotides. The yield is essentially quantitative if you get the right mix of the fragments. So if they're in the right stoichiometry and you have the process optimized, then the yield is very high. If you have a prochiral 5' thiophosphate as the substrate instead of a monophosphate, by virtue of the fact that this reaction is stereospecific, you get RP stereochemistry for the phosphorothioate exclusively in the product. And so that's an important consideration for antisense oligonucleotides where the ligation site may be a phosphorothioate linkage. And so let's take a look at the immediate applications for ligation. And siRNA first. This is synthesized by sticky end ligation, so-called. It was described by Gobind Khurana first in 1968. And in this method, it's very flexible. You can apply fragments of different lengths, different numbers of fragments, and develop a good process. So Alnylam and Almac together, they published on this, and their process has 6 fragments. We use 4. We just find that works best for us. And so we hybridize these 4 fragments, and then the sticky ends cohese through Watson-Crick interactions, and then the junctions between the fragments are ligated. The oligonucleotide is purified by non-denaturing HPLC, so we generate the duplex as the final purified material, obviating the requirement for purification of the single strand separately, which represents a significant process improvement. The single guide RNAs, meanwhile, can be accessed through splinted or template ligation, in which different numbers of fragments overlap with different numbers of templates, depending on the length of the oligonucleotide and your strategy. And these fragments, the junctions between the fragments are ligated, and the DNA is removed. Sorry, the templates are removed. So we use DNA templates, and so we can simply digest those DNA templates at the end of the reaction. But they can be removed through other denaturing conditions. And then the final single guide RNA that's purified by HPLC to generate the product. And here are some examples of products that we've synthesized. These top 2 molecules, these siRNAs, these are both GalNAc conjugates with chemistry that is typical of that you would find in molecules in clinical development or commercial development. These were synthesized by sticky end ligation. And of note, the 4 fragments that we used, none of them were purified by HPLC. So these are all UFDF processed material. And we're generating siRNAs that are 95 and 97% pure. That's with a denaturing UPLC method and data that's been verified by the customer. These, and that top one, that's currently in our GMP suite. These 2 single guide RNAs, one of them's a 100 mer, that's typical of the Cas9 nuclease type length. And then this 161 mer, that's more aligned with the length for a prime editing guide. So again, these are chemistries that are typical of those for molecules in clinical development. And these are synthesized by splinted ligation, and after removal of the template and purification, we generate material in 96 and 98% purity. And that compares with purities of about 80% that you would get using the exclusively generation 1 synthesis. This final example is interesting. It's a 44-mer, and it contains a phosphoramidate backbone linkage. And we synthesized that also by splinted ligation. It got 97% purity, and that compares with a purity of about 85% when this same molecule was synthesized at a different location using exclusively generation 1 chemistry. Some features of ligation. Firstly, the quality. So short-mer impurities are rejected. We always see higher purity compared to traditional generation 1 processes. And maybe this is because there's less opportunity for impurities to be compounded during the iterative synthesis. There's just less cycles for fragments. And then we see high yielding conversion because of the, if you get the stoichiometry right, it's almost quantitative. It's promiscuous to 2' ribose chemistry, tolerates phosphorothioate and other backbone linkages, and tolerates ligands such as GalNAc on either end of the molecule. It's modular, so we don't see any reason that this technology can't be adapted way beyond 160 mer. It's adaptable to batch mode and single-use bioreactors, which makes it scalable. And as we've seen, fragments don't always need to be purified. And we're investigating whether that may also be the case for API. And so we have great scalable methods for manufacturing by ligation. So the next thing to look at is how we make the fragments. And fragments, as are oligonucleotides in general today, these are manufactured using the phosphoramidite method, generation 1. We have these flow-through synthesizers. This is a picture of an oligoprocess synthesizer from our manufacturing suite. This is the upper end of scales that's available today. And in this type of system, the reagents flow through this column, and they flow through a bed of the solid support, either CPG or polystyrene typically. And there are some CMC challenges associated with this. Firstly, the scale. It's limited, in our hands at least, to about 7 kg per batch. And there's just, engineering, physics and engineering, there's just limits to the bed height and diameter of the column. Typically, the column diameters get up to about 1 meter. So with this type of setup, 1 ton of API would require scaling out. For 7 kg batches, that's about 140 batches. So scaling out as opposed to scaling up. And then raw materials, equipment, sweet time, those are all really expensive. So we shouldn't be surprised to see prices for 1 kilo of siRNA or 10 grams of single guide RNA approaching $1 million on the market today. Environmental sustainability, so it's a low atom efficiency, so there's a lot of equivalence of reagents that are required, and that leads to high process mass intensity, and this is less than ideal. Yield declines exponentially with oligo length, and meanwhile impurities accumulate with the oligo length, and so this is a problem that's compounded, particularly for longer oligos like single guide RNA. But there are some promising approaches that are being investigated to meet these challenges and scale oligonucleotide production. And going from left to right, solid phase oligosynthesis can be scaled by transitioning from the solid phase flow synthesizers to batch reactors. And this is the paradigm for the peptide field. Also, we have these liquid phase oligosynthesis systems, such as the hydrophobic supports or molecular hiving. Agiphase is the most famous of these, and this was approved for manufacture of an oligo drug in 2020. And then on organic solvent nanofiltration, where the oligonucleotide is grown on this hub, which is retained by an organic compatible membrane, which allows other reagents and small molecules to flow through. And there are other sort of newer approaches. Mechanochemistry is being pioneered in Massad's lab at McGill. And Biogen have synthesized fragments for the purpose of assembling them into ASOs. So we will get there. Future directions, we'll see continued optimization of ligation processes for siRNA and single guide, and we'll extend to other RNA modalities, including mRNA. There's no reason that can't be done, which opens up the exciting prospect of mRNA with discrete chemical modifications. We can expect to see antisense oligonucleotides being synthesized this way with control of stereochemistry. And we'll see optimization of the existing, and we'll see new fragment manufacturing methods. We'll expect to see convergence of computational chemistry and AI for better enzyme design. And if anybody doubts that, you can refer to the work at David Baker's lab at University of Washington for de novo design of enzymes. And then at some point, regulators are going to be asking drug sponsors why they're not manufacturing their oligonucleotide products using ligation technology, because the advantages are so clear. And so through that, we would expect to see wider adoption also. So to conclude, market demand for oligos is anticipated to grow significantly over the next decade, and this demand can be met by adoption of fragment ligation technology. It's ready now for sgRNA, siRNA, and other oligos. And the advantages are clear. They include improved quality, yield, environmental sustainability, and scalability compared to purely chemical processes. The fragments are currently manufactured using these scale-limited solid phase flow synthesizers, and that really will have to change, I think, if we're going to meet the demands for tens of tons of oligonucleotides in the future. And that'll be met through the adoption of scalable technologies that get us from these systems and into batch reactors. Some acknowledgements. I can't thank the phenomenal team at Hongene enough. But for the oligo team, that's led by Jim Yang, and the biocatalysis team is led by Minju Wei. I want to give a nod to the people at Hanson Wade too. They were really helpful in compiling the data for the graphs for the market data. And thank you to all of you for listening. And we have time for some questions, I hope. And I have some colleagues here this week, so please visit us, and look forward to talking to you more during the conference. So thank you.
Thank you so much. Questions, please.
Thank you very much. That was a great talk. If you look forward in the next 10 years, how long do you think it's going to be? How long do you think you can make messenger RNAs?
The length in terms of the number of nucleotides?
Yes, for a commercial purpose.
I don't think there is a limit. I think that the approach is absolutely modular. So I don't see any reason that it can't be extended to molecules that are certainly much longer than 160. There was a presentation from Ajinomoto at TIDES where they demonstrated, I think it was a 560 mer. But it's modular, so I think it can be extended much beyond that. Yeah.
David, that was a great talk. Congratulations to Hongene and everybody involved in this work. My question is rather granular. You know, as chemists, when we start from the phosphoramidite synthesis, we are able to scale it up 100 grams and something like that quickly, and the chemists make it. On the other hand, triphosphate synthesis is not there yet. You know, they are struggling to make in 10 milligram quantities, at least in my lab. So what is the best method? I mean, do you see that as a limitation in this ligation processes you are talking about, because that also involves chemistry, or there is an enzymatic method which will address which will overcome Eckstein's methods and so on? So two basic questions. What's the best method of making the triphosphate? Scalability, are there any alternate enzymatic processes?
Right, so we, for the NTPs would be required for the fully enzymatic process. Yeah. But you're exactly right, those are very early in development. They've been manufactured at scale for vaccines and other mRNA products, albeit without the 3' protection, which adds a lot of complexity. But the price for those, the cost for manufacture is still very high. It's orders of magnitude higher than it is for a phosphoramidite, I think. So the manufacturing processes, the methods need to be improved, and we're working on doing that now, but it is going to take time. It's going to take working with partners who are developing the enzymatic technologies to work together to bring those prices down so that they're more aligned with what we would see for phosphoramidites. Otherwise, that does present a very real barrier to entry for that technology into the market. But for the ligation technology, those are 5' monophosphates on the fragments, and we synthesize those chemically. You can synthesize those with enzymes, and that's the way that Alnylam and Almac reported. So that could be a process improvement that we would incorporate later on as well. But for now, for us, it's just easier to make those chemically.
David, thanks very much. So we see the advantages of ligation. Purity was quite clear there. What about scalability now, right? So you talked about the needs for multigrams and tons. So what are the challenges there for, I know you can make the fragments in large amounts, but now you're going to ligate, there is separations of enzymes, etc. So
yeah, it's actually the other way around. So yeah, we have the I think we have a scalable approach for manufacturing the oligo from the fragments. Albeit, there's things to work out there, right? Like how are things removed at scale? But the key thing is that can be adapted to a bioreactor batch type process. Whereas the manufacture of the fragments, that's the key part that we need to get away from the solid phase flow synthesizers and into a batch reactor type format, whether that is solid phase or liquid phase, or people develop both eventually, which is entirely possible. Then that's the key transition that needs to occur. Yeah.